Because I've been trying to split my talk in two parts. In the first part, I will aim to give you an overview of the current knowledge based on EEG sleep studies in humans to identify whether there are any sleep disturbances in major depressive disorders and whether any of the EEG extracted variables can constitute biomarkers. In the second part of the talk, I will show you a recent study using a rodent model of major depressive disorder to try to see whether we can establish a translational model of sleep disturbances. So depression is a common condition. A major concern is the fact that estimates of lifetime prevalence are ranging up to 20% of the general population. Major depressive disorder is characterized by affective, cognitive, and physiological impairments. And I've listed here some of the most common clinical symptoms. Interestingly, sleep problems are very often inadequately addressed in the clinic, although they are very well known to contribute to other of the affective disorders such as depressed mood, but also to the cognitive dysfunction, including deficits in attention and cognitive impairments. So those sleep disturbances are thought to be core symptoms of depression. They are actually part of the diagnosis defined by the DSM classification. It's actually quite important to say that a majority of the depressive patients do complain of sleep problems, more than 90% of them. They often complain of insomnia, and the subjective complaints is actually verified by polysomnographic studies, which have been shown that you have disturbances in sleep continuity. This is, for example, encompassing an increased difficulty to fall asleep, which is reflected in the EEG by an increased latency to fall asleep. There are also more frequent awakenings during the nights, which is associated with this difficulty in maintaining sleep. And you have also many early awakenings, and I will show you an example in the next slide. You also have insomnia, which is shown objectively by a decrease in slow-wave sleep. It's important because slow-wave sleep has been associated with the restorative function of sleep. And not only that, there's also a dysregulation of REM sleep, which is also known as paradoxical sleep. Why is it relevant or interesting in the context of depression? Is because REM sleep has been demonstrated to be involved in the regulation of cognitive processes, but also emotional processes and emotional memory. So this dysregulation is characterized by a shortened REM sleep latency, increased REM sleep duration, and an increased density of rapid eye movements that are characteristic of REM sleep or paradoxical sleep. So how does it look like? First, if you are inviting a subject in the sleep lab, you will be able to record from the EEG and the EOG, and you will be obtaining a typical sleep hypnogram. So this is the time course of sleep during an 8-hour opportunity to fall asleep. Here, for a healthy subject, you will see that the subject is awake, is rapidly entering shallow stages of sleep, then it's entering deep slow-wave sleep. Then you will have a cycle. It will reach rapid eye movement. Typically, it occurs after 70 to 90 minutes in a healthy patient, and you will see this very nice cycle of non-REM REM, non-NREM throughout the night, and you see that it's a very good sleeper because it's waking up quite late. In contrast, if you take a typical hypnogram of a depressed patient, you can first see what I mentioned in the first slide. You have this reduced latency to REM sleep, which is reduced to 50 to 60 minutes. You can also observe that you have far less slow-wave sleep in this patient, especially in the first non-REM REM cycle compared to the healthy subjects. You can also see that this patient has really problems to maintain sleep with all the rapid awakenings, and also this person is waking up quite early, around 4:30. Okay? So this is a typical image that you will see in depressed sufferer. Now, since the 1960s, it has been a graal to establish whether such EEG variables would constitute a biomarker. There's different ways of defining biomarkers, and in the sleep literature, it was actually proposed that some of those variables were constituting trait or vulnerability markers, meaning that they would be present before the onset of the depression, and that they would persist thereafter. Or it was proposed that some of those variables would be only state marker, meaning that they would occur during the acute phase and then would be disappearing under remission. Then some people proposed that some of those were representing scars, so only due to the disorder itself, and some other people suggested that some of those markers would represent endophenotypes. So I will spare you the hundreds of studies. There were many inconsistent findings, mixed results, but there is a very, before we go to that, because I will rebound on what has been said earlier, in terms of trait marker, EEG is actually quite valuable. So this is here a healthy subject. One subject is actually a healthy young university student who's been invited into the sleep lab 4 nights at 1 month interval. And this is here the traditional EEG spectral analysis using fast Fourier transform. You decompose your EEG into power density across into frequencies. And what is quite remarkable, you can see the stability of the EEG signal with the traces that are almost superimposable. At the same time, the EEG allows you to capture the inter-individual differences, and here you have 4 nights, and you have 8 subjects, and each of them are color-coded. Okay. Also, as shown this morning, I wanted to highlight the fact that the quantitative EEG analysis allows you to discriminate between different drugs and their mechanism of action. We heard earlier about zolpidem, the gold standard of hypnotics, with the very well-known benzodiazepine fingerprints on the EEG, with a reduced EEG power for frequencies below 10 Hertz and an increase in the spindle range. And in this can be actually compared, or to be differentiated from a different mechanism of action, gaboxadol, also known as ZIP, which is also acting on GABA A receptors. Not only you can discriminate between drug effects, but you can also identify sex differences. And the differences between, you can see here that women have a larger response to both gaboxadol and zolpidem, and this is actually quite interesting to use to identify which doses should be given to healthy subjects as well as patients. And this is translational because you can actually reproduce those EEG signatures of the 2 different drugs also here in mice, but that also has been shown in rats. So if I go back to the biomarkers, there's been a recent meta-analysis looking at 886 EEG sleep studies performed in depression. Only 56 studies were selected, and we can maybe discuss afterwards why. But what they were able to show is quite interesting, is that there are very significant differences in several EEG parameters between major depressive disorder and controls. I have listed them here, and in the order of from the largest size effect to the smallest size effect. Interestingly, those are all the parameters that I mentioned before, and some of them have been shown to be moderated by depression severity. Only 2 parameters were not altered by the severity of the depression, and this is REM, rapid eye movement density, and the duration of slow wave sleep. Now, if you look, so all those could be constituting state markers. Now, if you compare a major depressive disorder versus patients that are under remission, all those parameters have been shown to be significantly different. All of those are actually normalized under remission, meaning that people no longer show those changes. There's only two parameters that show no differences. This is REM density and slow wave sleep when the meta-analysis was aggregating all the studies together. Now, if you want to know whether REM density and slow wave sleep could represent endophenotype, you need to be looking at never depressed high-risk probands, so first-degree relatives of people suffering from depression, and compare them with controls. And again, the only two EEG parameters that are showing up as significantly different are the rapid eye movement density and slow wave sleep. So it has been proposed that many of those variables were actually representing state markers, but REM density and slow wave sleep could have a predictive value and are true endophenotypes for sleep in depression. Now, the reason why it's important is because there is really a need to develop new antidepressant drugs, in particular because 1/3 of depressive patients do not respond to current pharmacotherapy. Okay, so this was discussed this morning. Many of the drugs that have been developed failed, and the current problem here is in preclinical model is the ability to emulate not only depressive symptomatology, but also the treatment response. Okay? So we are focusing in the lab in sleep disturbances because they are inadequately addressed in depression. It's well established in the literature that those sleep disturbances are an increased risk factor for the onset, but also for the recurrence of depressive disorders. They are associated with poorer clinical outcomes and also slower and lower rates of remission. Okay. Those sleep disturbances have also been shown to have a negative impact on treatment response. And in contrast, what is positive is that when a treatment of insomnia has been combined with antidepressant, it has been shown to improve the symptoms of depression. Okay. So, but one of the major questions which is not answered with human studies is when do the sleep disturbances arise with the development of the depressive disorder? And this is what we are aiming to do. Can we establish a translational model of sleep disturbances in depression? So our primary objective is to actually document the emergence of sleep and also circadian rhythm in a rodent model of major depression, focusing on the gradual development of the depressive-like states for our model, then study the time course of those sleep disturbances and compare them with the emergence of other depressive symptoms such as behavioral or physical symptoms. We choose the following rodent model of depression, the unpredictable chronic mild stress paradigm, for two reasons. The first one is because it's considered by many to be one of the more useful model to actually assess antidepressant activity, and also because it's a chronic model and it allows you this gradual development of the depressive-like states. So how does this work? This is a naturalistic model. You are applying every day different social or environmental low-intensity stressor, and this is a very important aspect. There are no major or mild stressors, and they are applied in the randomized way because you do not want animals to habituate. So you need to make sure that it retains this unpredictable characteristic. And we have here some cartoons to show you some of the stressors that we are using. So we have two experimental groups, the unchallenged control group, which is constituted of 9 mice, and we have the daily UCMS challenge group with 9 mice. And in this current protocol that we use a very long paradigm lasting more than 2 months. Okay? And then we measure different outcome variables. First, we need to demonstrate that this model is actually a good model for depression. So I will show you some results on physical and behavioral measures. We also assessed physiological parameters such as the stress HPA axis. We looked into the locomotor activity throughout the 2 months protocol, but because of time, I won't be able to show you some of this data. And I will focus on the EEG study, where we perform 24-hour EEG, EMG recording every week of the protocol to see the occurrence of the changes. So first, if you focus on physical assessment of the depressive-like states, it's actually quite easy to establish in an animal model. The body weight, so rodents are increasing weight with age. So the way we color-coded, the control group is blue and the UCMS depressive group is red. And while the control group is showing this steady increase in body weight, you see that after baseline, the UCMS group failed to gain weight at the same speed as the control. Another very interesting aspect of physical assessment of depressive-like states in rodents is associated with coat states. So a normal animal will always have this very nice fur because of the grooming behavior, spontaneous grooming behavior, while an animal which is developing a depressive-like state will stop grooming as much, and you see this deterioration and worsening of the coat state and weakened maintaining. So it's emerging after 2 weeks of UCMS, and it can remain steady. You can also further look at self-centered behavior by trying to see whether you can induce grooming behavior. And so we are spraying a 10% sucrose solution and looking at how fast the mice is starting to groom, to lick away the sucrose. You can see that the control group is quite fast, while the UCMS group is increasing its latency to lick. And you can also see that the grooming duration is significantly reduced in the UCMS group. Now, another way to measure motivation in animal is actually looking at whether they do a nice bed for themselves. So you give them a cotton nestlet, and a normal animal will be very motivated and will shred the cotton nestlets and do this very nice one. Okay, so you have a score of 5 when you do a very nice bed, and when the cotton nestlets remain untouched because the animal is not motivated, then you have a score of 1. You can see that the control has a steady 4.5 to 5 maximal score, while with the UCMS you have a reduced motivation towards the third week onwards. Depressive and anxiety-like behaviors can be actually modeled with different tests. So here we have the acute aversive force swim test, which is based on aversion of the animal, and the despair of the animal is associated with an increased latency to be immobile. Another typical test to evaluate depressive and anxiety-like behaviors is the novelty suppressed feeding, where there is a conflict between the desire to eat a palatable, attractable food, which is a cookie, versus the fear of entering an open and brightly lit space. And one can see is that the depressed group is actually taking much more time to go to the food. And finally, if you look at aggression or social behavior, one can look at the resident intruder test, and one can see that the depressive-like test, the depressive-like group is actually showing more aggressivity by reduced latency to attack the intruder, and also by the increased number of attacks. Okay, so it's a little bit tedious, but it is absolutely key to demonstrate that we do have a depressive group using this paradigm. And finally, if you want to assess the stress axis, so the hypothalamic pituitary adrenal cortex axis is responsible for our neuroendocrine response to stress. And this stress response is stopped by a negative feedback loop. In depressed patients, it has been shown that you have an increased release of cortisol, so an increased release of glucocorticoid, which is due to this to an impairment of this negative feedback loop, and this can be shown using the dexamethasone suppression test. So if we do the same thing in animal, we can show that while dexamethasone is an agonist of glucocorticoid, and you can see that dexamethasone is able to steadily suppress throughout the 9 weeks protocol the release of corticosterone in control, while it's no longer feasible after 2 weeks onwards by the depressive group, which is also mimicking. So focusing now on the alteration, we were being able to show that although at baseline the two groups were exactly the same, you see a steady decrease in slow wave sleep in the UCMS group, and this is seen both during the dark active period but also in the light active periods. So this is mimicking insomnia observed in depressive patients. This is here, you can see that the non-REM sleep episode length is also decreased in the depressive group, and the number of count is increasing, which reflects a fragmentation of sleep in this model. In contrast, for paradoxical sleep, as observed in human in the depressive state, you can see an increase which starts right after 3 days of the UCMS paradigm, and this is shown both for the dark and the light period. Same thing for the episode length. You have an increase in the REM sleep bout, and you also have an increase in the REM sleep bout count. So you have an increased REM sleep continuity. Now, if we focus on EEG power, I represented here 100% baseline, and you see the modification. So you can see that in blue, the control and the UCMS, there are no differences in non-REM sleep EEG power, which is also corresponding to what you see in males, in men in depressive patients. In contrast, it seems it's a preliminary analysis of EEG power density in REM sleep. It seems that very early on there is this increase in the theta peak, which is characteristic of REM sleep and which can be observed from week 1 to throughout the protocol. Why is it important? Because again, paradoxical sleep has been proposed to be associated with the consolidation of memories associated with negative valence. So in conclusion, this UCMS paradigm is able to emulate many different symptoms of the human major depression, and in particular the sleep disturbances, because we are able to recapitulate most of the sleep disturbances observed in human. And what was very striking to us is the fact that those sleep disturbances are actually emerging right after 3 days of the paradigm, and they are sustained throughout the protocol, and they are actually occurring before the impairment in self-centered behavior, weight loss, loss of motivation, or the changes in the HPA axis. So we believe that we have a promising translational model of sleep disturbances that we can use. We will use it to further investigate the emergence and the time course of the sleep abnormalities, but it could be used for the characterization of novel antidepressants. Now, this work has been sponsored by Lilly. All the whole experiment, and it's an Herculean effort, has been performed by Mathieu Nollet, a postdoctoral fellow, who is working together with me and Dakian Dijk at the University of Surrey within the Surrey Sleep Research Center, but also at Lilly with Keith Wayford, and Andy, who is in the audience, has been also contributing to some of the sleep data analysis. Thank you very much.
Thank you. Perfect. Time for a couple of questions.
So the behavioral and the physical symptoms will be normalizing gradually. In terms of sleep symptoms, this is our next experiment. So if we trust the meta-analysis, you could predict that after ceasing the protocol, you could have maybe a normalization in the slow wave sleep duration, in the REM sleep duration. Rapid eye movement density, we do not measure. It's very tricky to actually record at the level of the animals. So, but we would expect a normalization of most of the changes that we have seen for those that we can measure.
I've got a question. So your endophenotypes are really interesting in the human sleep and in depression particularly. And you mentioned that in probands.
Yes.
So those are very closely related relatives.
Yes. So those are first-degree relatives.
So does this suggest that there's a genetic component then to this?
Yes.
And then how much do we know about the genetics of depression?
So there is a proposed genetic components. I didn't have time to say, but in those high-risk proband study, it has been shown that if you measure the same individuals 3 years and a half after, all of them do have still the changes. And it seems that, well, there is a suggestion that it would be associated with some of the cholinergic receptors, but also potentially with circadian rhythms genes. So, for example, clock or BMAL or PER3, but not much is known.
But they haven't shown up in any genetics associated studies in depression yet.
PER3 came up, clock as well.
Okay.
So some of the circadian rhythms.